Along with advances in DNA technology, scientists have been developing and refining methods for cloning whole multicellular organisms from single cells. Cloning produces one or more organisms that are genetically identical to the parent that donated the single cell. This is often called organismal cloning to differentiate it from gene cloning, and more significantly from cell cloning. Cell cloning is the division of an asexual reproducing cell, such as a bacterium, into a group of genetically identical cells. In fact, the word clone comes from the Greek klon, meaning twig. The current interest in organismal cloning arises primarily from its ability to generate stem cells. Stem cell is a relatively unspecialized cell that can both reproduce itself indefinitely and differentiate into specialized cells of one or more types. Next is the brief history of cloning. The cloning of plants and animals was first attempted over 50 years ago in experiments designed to answer basic biological questions. For example, researchers wonder, is it all the cells of an organism have the same genes, or whether cells lose genes during the process of differentiation? One way to answer this question is to see whether a differentiated cell can generate a whole organism or not, or in other words, whether cloning an organism is possible. So, let's discuss these early experiments before we consider more recent progress in organismal cloning and procedures. So, there are two types of early experiment in organismal cloning. The first one is cloning plants, which is the single cell cultures, and the second one is the cloning animal, nuclear transplantation. The successful cloning of whole plants from single differentiated cells was accomplished during the 1950s by F.T. Stewart and his student at Cornell University who worked with carrot plants. They found that the differentiated cells taken from the root and incubated in culture medium could grow into normal adult plants, each genetically identical to the parent cell. These results show that differentiation does not necessarily involve irreversible changes in the DNA. In plants, mature cells can de-differentiate and then give rise to all the specialized cell types of the organism. Any cell with this potential is said to be totipotent. In fact, you yourself may be a plant cloner if you have ever grown a new plant by cutting. Then you have practiced cloning. Plant cloning is used extensively in agriculture. For plants such as orchids, cloning is the only commercially practical means of reproducing plants. The next early experiment that have done by the researchers towards the animal, which is by the nuclear transplantation.
Such experiments were conducted on one species of frog named Rana pipiens by Robert Briggs and Thomas King in 1915, and on another frog species, Xenopus laevis, by John Gordon in the 1970s.
This procedure is called as the nuclear transplantation and now more commonly called as the somatic cell nuclear transfer. So their approach was to remove the nucleus from an egg, called as the enucleated egg, and replace it with the nucleus of a differentiated cell. So next is the reproductive cloning of mammals. In addition to cloning of frogs, researchers were able to clone mammals using early embryonic cells as a source of donor nuclei. In 1997, researchers in Scotland announced the birth of Dolly, a lamb clone from an adult sheep by nuclear transfer from a differentiated mammary gland cell.
Dolly is the first animal that was created by cloning in the science lab. The researchers implanted early embryos into surrogate mothers. Out of several hundred embryos, Only one successfully completed normal development, and it was Dolly, a genetic clone of the nucleus donor.
Since that time, after the Dolly, researchers have cloned numerous other mammals including mice, cats, cows, horses, pigs, dogs, and monkeys.
In conclusion, the researchers stated that cloned animals of the same species do not always look or behave identically. In a herd of cows clones from the same line of cultured cell, certain cows are dominant in behavior and others are more submissive. Another example of non-identity in clones is the first cloned cat, named Sissy, stands for carbon copy. She has a calico coat like her single female parent, but the color and pattern are different because of random X chromosome inactivation, which is a normal occurrence during embryonic development.
Have you ever wondered, what does cloning do today?
Imposing a full lockdown on Kuala Lumpur and 5 states to curb a third wave of COVID-19. The 2-week lockdown begins on Wednesday. Interstate travel will also be banned nationwide.
Stay at home.
Prime Minister Muhyiddin Yassin says that at the current rate of infection, daily cases could rise.
From March 2020, the world is facing with the crisis of pandemic from coronavirus. The virus that causing the COVID-19 is known as Severe Acute Respiratory Syndrome Coronavirus 2, or in short as SARS Coronavirus 2. The virus spread through the droplet that comes out from nose or mouth. The researchers have done several studies on producing monoclonal antibodies which apply the technique of cloning.
What is the monoclonal antibodies? The word of monoclonal means a single clone. Monoclonal antibodies are identical antibodies produced in laboratories from single of immune cell. The clone of immune cell are genetic completely identical, and so the antibodies that produce also identical to another which perfect work properly with large number. Cloning technology, which focus more on monoclonal antibodies, have an ability to help and guide in the vaccine production. Instead of vaccine or other medicine itself, monoclonal antibodies are also need to be highlighted as one of the treatment. Monoclonal antibodies is an antibody that was created by cloning the relevant antibodies from human. Up to 2020 until now, the world is facing with the crisis of coronavirus disease COVID-19. SARS coronavirus 2 is an agent that was identified as a factor toward COVID-19. Scientists have isolated monoclonal antibodies from the patients, commonly from the B cells, who have recovered from the COVID-19, while, in some cases, it is obtained from the patient who were infected with respiratory syndrome coronavirus to be neutralized it later.
The researchers discovered that SARS coronavirus 2 neutralizing monoclonal antibodies is the surface spike glycoprotein that provides the entrance of the viral into the host cells. The interaction between the surface spike and the ACE2 receptor, which found on numerous cells, causing to the viral infections.
Thus, by neutralizing the monoclonal antibodies, will block the infections. Since the coronavirus is rising, more than 70 monoclonal antibodies product was produced from various companies. In conclusion, the researcher stated that monoclonal antibodies by cell cloning is able to prevent and treat early infection of COVID-19.